Well, thank you. As you can see, I'm not Mike Ditka, and it's a bit of a challenge to come before an audience expecting to see a very famous football coach, a newscaster, and just a very inspirational individual. And so you might be asking yourself, why would they ask this professor from Texas to come here and substitute for Mr. Ditka? And the answer is he and I go way back. We've actually have been long-term friends. Before he got into football, he actually was in a different league that he and I shared together, and I just thought I would show you that. We were both teammates on the Gladiators. That's my humor. So anyway, hopefully what I'll be able to do in the next 30 minutes or so is to entertain you in a different way by discussing the role of telomeres and telomerase and its relationship to aging and cancer. So by way of introduction, I do have a disclosure that I am a scientific advisor to LifeLength, a Madrid-based company involved in telomere length testing as a biomarker of biological versus chronological aging. So with that behind me, I just want to remind everybody, I think it's very obvious that today, as opposed to maybe 50 or 100 years ago, when we didn't have a protected environment, when most people, in fact, died of different types of infectious diseases and things such as that, because we didn't have antibiotics, we didn't have good sanitation, we didn't have all of the wonderful biomedical interventions. So now that people are living longer, the question is, what really does limit the maximum lifetime of individuals? And what I hope to share with you today is some of our basic science and some of our clinical experience trying to work on this one field involving the ends of our chromosomes called telomeres. Now, this actually is a field that's been around for a long time. It was actually in the 1930s and 40s that both Dr. Muller and Dr. McClintock both received Nobel Prizes for basically what I show at the bottom of this slide, about describing the ends of all of our chromosomes. So all of our genes are on chromosomes. We have 46 chromosomes. Each end is different, and there are these repetitive elements at the ends of all these chromosomes. They don't code for genes, and these two scientists that each got individual Nobel Prizes were critical in showing that there were special complexes at the end of the eukaryotic chromosome. It turns out that these structures were termed by Dr. Muller telomeres. And what's interesting is all vertebrates have this same sequence at the end of all of our chromosomes. There's this sequence TTAGGG, and these are monotonously repeated thousands of times. And another Nobel laureate by the name of Jim Watson, who discovered DNA, was the first to show that the phenomena was called the end replication problem, that every time a human cell divides, your telomeres get a little bit shorter. And this is due to the mechanism by which DNA replicates. And there's other things. There are environmental aspects like oxidative damage and other end processing events that can also lead to increased losses at the ends of our chromosomes. And the hypothesis that came out about 20 years ago is that senescence, or aging, occurs when some of these telomeres get very short. So what is the data? So how do you think about telomeres? Let's just give an analogy here. So everybody knows what the little plastic tips on the ends of shoelaces are called, right? Auglets. And they're covered these little plastic things. So you can think of your telomeres in all of your cells in your body as having, when you're young, good protected plastic tips. Now what happens as you get old, a subset of those plastic tips become unraveled. And when a tip becomes unraveled, or a chromosome becomes unprotected, that leads to a growth arrest of that cell, and that cell then to no longer divide and sustain and replace the tissues in the body. So the data actually evolved almost 60 years ago with Leonard Hayflick, who was at the Wistar Institute in Philadelphia. And Dr. Hayflick overcame a long-standing dogma that cells extracted from the human body were in fact immortal. That was the thought collective in the early 1950s. His experiments clearly showed, it is illustrated in this slide, that in fact normal human cells taken from the human body actually have a limited capacity to divide, and that when the cells reached a certain number of doublings or passages, the cells would look like here on the right. They would get big, and they would actually secrete a whole different constellation of proteins that's quite different from what you would see in a young quiescent cell. And then in 1990, a group published the first evidence that it was the shortening, the gradual shortening of telomeres that correlated with aging in a test tube or in vitro. Now, of course, there's a lot of correlations in science, but it certainly didn't prove cause and effect, and maybe this was only something in tissue culture which may not be replicated all the way into human beings. So at the same time, another group actually started measuring telomere lengths in real human tissues. And as you can see on this slide, that if you look at fetal or reproductive tissues, that telomeres in humans are somewhere between 15 and 20 kilobase pairs at the ends of our telomeres. And as we get older, in all of our cells that divide, you see gradually shortening of telomeres. And it increases basically throughout life until when you're in your 70s or 80s, most individuals have telomeres in the very critically short amounts.